Very good morning to all the NEET aspirants. This is your Prasanna Ma'am from NEET World. So yes, we are in the questionnaire of the chapter Plant Growth and Development. So here today we are going to exercise both the developmental patterns, heterophily, plasticity type of questions, and the questions on differentiation, dedifferentiation and redifferentiation. The next questionnaire session after this will be on the phytohormones. So before getting into the questionnaire, I would like to give you a brief introduction about the special programs which are being held in the NEET World, Narayangada, Hyderabad. So here we have 2 Steps to Medicine program for 11th standard students and NEET World's 1 Step to Medicine for 12th standard students, NEET World's Power Step to Medicine for the students who are aspiring to join in long term. For all the 3 special programs, the fee is very minimal, which is just 3333 per month with complete study material and 2 master revision strokes after the completion of the syllabus. To know more about these programs, you can call us to the given number or you can directly visit our branch which is in Narayangada, Hyderabad. So yes, let's get started with the questionnaire. First, dash are the examples of tissues formed by dedifferentiation. Dedifferentiation means those cells which have lost their capacity to be permanent, okay, those cells which regain the capacity of meristematic activity. Amongst these options, you tell me which are those cells which have regained the meristematic activity. Interfascicular cambium, correct, and cork cambium. These 2 are the tissues which have regained the meristematic activity. So here both option A and B are correct. Look into the second question beta. Examples to tissues that are formed by redifferentiation. What are the examples below? Those are formed by redifferentiation. That means after the meristematic activity, the dedifferentiated tissues meristematic activity, those cells which are again getting permanent are called as redifferentiated tissues. So here redifferentiated tissues can be what ma? Secondary cortex, secondary phloem and cork cells which are also called as phellum, right ma? So here the right answer will be option 4, all of this will be the right answer. Next, look at third question keenly. Yeah. What would be expected to happen if dividing cells stop differentiating? Once dividing cells will stop differentiating, what would be happening over here? That is the question. Dedifferentiation activity takes place, okay, redifferentiation takes place. So here there will not be any possibility of dedifferentiation and redifferentiation because the dividing cells are stopping their activity. At that point, callus is formed, okay. What is formed? Callus is formed over here, okay. When the dividing cells, primary dividing cells or meristematic cells will stop their dividing activity, initially an undifferentiated mass of cells, that is callus is being formed. So here what will be the right answer? Option 2. Next question. Which of the following is an example of differentiation? Okay, amongst these 4 examples we need to choose which of the following is clearly explaining the differentiation activity. That means primary meristematic cells are getting permanent, okay, that is forming simple permanent tissues or complex permanent tissues, be it primary xylem or primary phloem. Look into this properly. The formation of tracheiary elements, xylem, tracheids and vessels. Yes, xylem, tracheids and vessels, primary tracheiary elements, that is because of what? Differentiation activity only. Formation of, yes, you can ask your doubt ma, just type it in the chat box, I will be answering you, okay. Yeah, option B, formation of meristem like interfascicular cambium from fully differentiated parenchyma cells. So this is the activity of dedifferentiation, yes or no? Option 2 is what? Formation of meristem like interfascicular cambium from fully differentiated parenchyma cells. This formation of interfascicular cambium is what activity? Dedifferentiated activity. Dedifferentiated. Clear, right? Next, formation of meristem like cork cambium from fully differentiated parenchyma cells. Okay, formation of meristem like cork cambium and fully differentiated parenchyma cells. This is also speaking about what? D-differentiation only. So it cannot be B and C. So we can eliminate option D as well. So what is the accurate answer over here? Formation of tracheary elements, xylem, tracheids and vessels will be the primary function of what? Differentiation activity. Next question you look into beta. Assertion and reason type of question, which is the most challenging type of question for all the students. Okay, so look it with keen interest. In assertion he is saying like differentiation is a process in which plant undergoes maturation, correct, right? Differentiation is a process in which plant is undergoing maturation. In reason which is provided over here is during differentiation cells will lose protoplasm. No, not all cells will lose protoplasm, only tracheary elements that we have looked into the previous question, right? Tracheary elements like tracheids and vessels and few sclerenchymatous tissues only will be losing their protoplasm at the time of differentiation activity. Not all the cells will be doing the same. Okay. Okay, so here during differentiation cells, not all cells will lose their protoplasm. So here reason will be wrong. In this question assertion is true and reason will be false. Okay, so here assertion is true and reason is false. Option C will be the right answer. Next question you see Vita. All changes that an organism goes through during the life cycle from seed germination or germination of the seed to senescence, all the processes or the changes that happen in a plant from seed germination to the complete development is called to the complete senescence is called as development. Okay, that complete phenomena is called as what? Plant development from seed germination to senescence. Very simple question. Look at this. In some plants the leaves of juvenile plants are different in shape from those in a mature plants. Okay, so the leaf shape will be different in the same plant at the juvenile stage and at the mature stage. This phenomena is called as what? Heterophily. This phenomena is called as heterophily due to phases of life. So it is because of the phases of life, right? So this is called as intrinsic development or intrinsic heterophily. Heterophily is one example of plasticity. Okay, so this is intrinsic heterophily that happens within the plant. Two different shapes of the leaves are observed, one in the juvenile phase and one at the mature phase, one type of shape in the mature phase. So here if you see what are the examples which show different leaf structure in juvenile and mature phase with respect to phases of life, phases of the plant life. So here the right options will be cotton, coriander and larkspur. These are the examples which show intrinsic heterophily, not buttercup. Okay, buttercup is with respect to habitual heterophily. So the leaf shape will be changed based on the habitat in which it is growing. It is not because of the phases of the life. Okay, so here the right answer will be what beta? Option D. Clear, right? Next question. In aquatic plants, Ranunculus flabilaris, submerged plant, that is nothing but the scientific name of buttercup. Submerged leaves are highly dissected, whereas emerged leaves are broad and lobed. Okay, emerged leaves will be broad and lobed. So this is an example of what? Again, heterophily. Heterophily is the example for this, but what type of heterophily? This is environmental plasticity. Environmental heterophily or environmental plasticity, one and the same. So here we can go with two options, that is both A and B. Option D will be the right answer over here. Next question. Look at this images. The following figure shows juvenile and adult, two different types of leaves at two phases of the life of a plant. So this is the heterophily in larkspur. Yes, correct. In larkspur we will see different shapes of the leaves based on the phase of the plant development. So here option A will be right. Heterophily in buttercup, no. In buttercup though it is based on the habitat. Heterophily in cotton and coriander, this is also true. the complete answer would be heterophily in cotton, coriander and larkspur. Option D will be the right answer. So here if in the question he is asking you except, exception, then we have to go with the option B. If he is asking you what are the complete set of examples which can define this figure or which can define this picture, then you have to go with option D. Clear, right, ma? Next question you look into, which of the following is an example of plasticity? So today I have placed the complete questions on heterophily and plasticity only. Very simple type of questions, not in an advanced level. Just keep on exercising this, okay? Which of the following is an example of plasticity? Heterophily in cotton, coriander and larkspur, yes, this is an example. This is intrinsic plasticity. Whereas heterophily in buttercup, this is extrinsic or habitual or environmental plasticity. So both of them are the examples of what? Plasticity only. So here the right answer will be option D. Both A and B are correct. Next, the following figure will show, see, this is showing the environmental plasticity in terrestrial habitat and in the water habitat. So here you look into the following figure will show heterophily in larkspur. No, in larkspur there is no habitual plasticity or heterophily. Heterophily in buttercup, yes. So here option B is right. So this is showing heterophily in buttercup. Next, heterophily in cotton and coriander. No, here also it is with respect to phases of plant life only. And option D, heterophily in cotton, coriander and larkspur, this is also with phases of life. So here the right answer will be option B. Okay, this is habitual plasticity. Best example or the only example is what, ma? Buttercup. Next, intrinsic heterophily is found in, okay, intrinsic heterophily is found in all except, okay, intrinsic heterophily, he is speaking about phases of plant life. It depends on the phases of the plant life, okay. Intrinsic heterophily based on the phases of the plant life can be cotton, buttercup, coriander, larkspur. So what are the examples for intrinsic heterophily or intrinsic plasticity? Cotton, coriander and larkspur. So here all except he is asking, except, so the answer will be buttercup, okay. Next, assertion reason type of question. Development is the sum of growth and differentiation, correct only. Development is sum of growth and differentiation. In reason, development in plants is under the control of extrinsic factors only. It is wrong, because development is not just under the control of the extrinsic factors, it is also under the control of the intrinsic factors. That is why we will be learning two types of factors, right, extrinsic and intrinsic, and examples as well. So here assertion is true and reason will be false. So assertion is true and reason is false. Option C will be the right answer. Yeah. Last question, if I am not wrong. Yeah. Last question, ma, assertion and reason type of question again. The difference in shapes of the leaves produced in air and those that produced in water in buttercup represents heterophilous development due to environment. So this is correct only. In reason, the phenomena of heterophily is an example of plasticity. Even this statement goes correct, but then both assertion and reason are true, but here in this assertion reason, nowhere the reason is explaining the assertion, right, ma? So here what option we have to choose? Both assertion and reason are true, but reason is not the correct explanation of assertion. So here option B goes good with this question, okay. So this is a basic questionnaire session. In the next session, we will be exercising the advanced questions on the phytohormones. So don't miss that questionnaire session tomorrow, okay. In the same time, once again, before winding up the session, I would like to give you a brief intro again about NEET World special programs, where we have 2 steps to medicine for 11th standard students who are aspiring for NEET 2027, and 1 step to medicine for 12 students who are aspiring for NEET 2026. Power step to medicine for long term students who are aspiring again for NEET 2026. any of the special course with the same minimal fee of 3333 per month. To know more about these courses, just call us to the given number, or you can directly visit our branch Narayanguda, Hyderabad. And one of the best advice for all the students who are planning for self-study to crack NEET 2026, NEET World is here with you with our Telegram channel. Okay, so join this Telegram group wherein you will be having daily quiz sessions of biology, chemistry, physics and mathematics who are aspiring for JEE also, and live YouTube class sessions will be updated here in this Telegram group, and free resources of study materials, questionnaire banks and question banks will be provided. One-on-one doubt solving sessions with the faculties are also available, where faculties will be directly addressing your doubts. Okay, and to know more about this, you can call us to the number, or to directly log in into this group, you can scan this QR code or directly visit our official website www.neetworld.in. Okay, that's all for today's session, and thank you. We'll meet in tomorrow's section with the advanced questionnaire practice of phytohormones. Thank you.